Hello, welcome everyone back to Potavirus. This is our fourth episode. So I'm Jessica and I'm here with Joe Campbell and Gina Suh today. So this is a special episode and I'm going to pass it over to Joe to kind of introduce a new phage therapy series that we have plotted to do. And so take it away, Joe.
Yeah, my name is Joe Campbell. For a long time, I was a program officer at NIAID, and I was running the bacteriophage interest group. And Jessica and I got to know each other while I was doing that. And Jessica and I have been interested in exploring the potential of the phage therapy with people who are intimately involved in it, and trying to figure out what's potentially keeping us from getting to that potential. And so we wanted to have a bunch of conversations with people who are experts in phage therapy. And our first guest is Gina, and I'll just let Gina briefly introduce herself, and then we'll start with a few questions.
Yeah, go ahead, Gina.
Great. I'm really excited to be here and invited by two of my most favorite phage people, Jessica and Joe. My name is Gina Suh. I am an infectious diseases physician at the Mayo Clinic, and I have been lucky enough to be able to treat patients, so far 17 treatment courses, with phage therapy.
Wow, 17 already.
It's 15 patients, 17 treatment courses.
Okay. Wow.
I guess we'll start out. We have a first question for you, which is when and how did you start getting involved in phage therapy?
Well, so I have been a clinician, so I see patients every day, all day, since, I guess, I don't know, for the past 15 years or so. And my subspecialty is orthopedic infectious diseases. And so that means that I treat patients with musculoskeletal infections such as osteomyelitis, so bone and joint infections, osteomyelitis, prosthetic joint infections. And while treating those patients was incredibly, gratifying, and we really made a lot of good progress and cured a lot of people, there were a subset of people that we just couldn't help because their infections kept coming back over and over and over again, no matter what we did. And it was really heartbreaking a lot of times. And we saw people's lives get ruined because they had an infection and they ended up not being able to walk or with an amputation. And so that really inspired me to kind of think about things that were different than what we were already doing. And that was at Stanford University, where I worked previously. And at the very same time, I just happened to start working with somebody there named Paul Boyke, who does a lot of basic science and translational, not necessarily phage therapy at the time, but not lytic, temperate phage work. And he wanted me to collect a bunch of wound samples for some experiments that he was doing. And so because I took care of wound infection patients, I did that. And then slowly I started to learn a little bit about these things called phages. And I was intrigued and started to kind of think about phage therapy. So this now would have been about 2016, 2017, and 2018. And then after that, I was recruited to Mayo for my ortho ID interests. And while I was there, in the first 6 months, we had a patient who eventually became our first phage therapy patient. But he had a recalcitrant, intractable infection of his prosthetic knee. And he approached us about phage therapy, not really even knowing that I was interested. And then one of our colleagues referred him to me. And, I looked at the case and I thought, this patient has, 10, 12 years of infection. They've been recurrent. There are about 3 or 4 or 5 bugs. He's allergic to everything on the face of the earth in terms of therapeutics or antimicrobials. there's no way this is going to work. Is what I thought. But instead of kind of saying, no, I talked to my colleagues and particularly my boss at the time, Dr. Barbari, who is our division chair. Dr. Barbari said, just go for it. At the very least, we'll have learned something. So I did that, and that patient is Mr. John Haverty, who we all know. His name is, public and published. And so John became our first phage patient. And then after that, it was just like a no-brainer. It was just like, it worked so well for him that after that, it became my purpose or calling, if you could say that, to keep going with this, because it changed his life.
So yeah, that story is incredible, and I remember following John Haverty's case, and I feel like I've met him, but I think I probably haven't. It's probably just been on webinars and seeing him speak online with you. Or yeah, no, we did a webinar together with you too. Yeah. Yes, that's what it's from. And so what have been the biggest changes in how you approach phage therapy now compared to in John's case?
I am much more careful about patient selection. So I select patients now with fewer variables and confounders. I look at things like how long have they had their infection, how many organisms are involved, how many isolates or sub-isolates are involved. If they have, 7, 8, 9, 14 different sub-isolates of Pseudomonas, that's not probably a great candidate. And in retrospect, I think maybe if John had come along now, I don't know that we would have necessarily have even treated him. And he was evaluated by other phage centers, and his case was not accepted. And so initially, and that's how he came, to knocking on our door, is after he had been to other places. And so I'm more careful about patient selection. I try to select patients who are both, in dire need, of course, but not patients who I think are highly likely to fail, because we want to learn, of course, but we also want to help patients, and with each, it's not easy, doing one of these courses. It takes a lot of time, and we have to select, 3 or 4 that we do in a year. So we have to be pretty selective. I also have come to respect pharmacokinetics a lot more. I know that the PK is something that we don't know all that much about with phage, and I am way more deliberate, I guess, in choosing a route of administration, and really trying to think through that, and also trying as best as I can to make decisions based on evidence or based on at least some kind of group consensus or something, because the stakes are high, I think, for not just the patient but the field. I want the field to move forward, and I want to be able to contribute with lessons learned or with successful cases, or both.
I just have a quick follow-up. Just wondering, like, when you say you wouldn't have potentially chosen to treat John if he came along now, like, what would be the main reason? Just because it is more difficult to find phages against that many isolates, or were there other reasons?
Primarily because he has a polymicrobial infection. So he had streptococcus, he had coagulase negative staph, he had enterococcus and Klebsiella. and maybe even more than that. I just can't remember off the top of my head. But yeah, the polymicrobial nature of John's infection maybe would have deterred us. And I don't know to what degree that impacts. I mean, for John's case, initially it didn't impact our success because we were able to treat the Klebsiella, and he had a very dramatic improvement in his symptoms. But 3 years later, he did come back with an enterococcus infection. So the enterococcus that had been in the joint from years ago reappeared, despite the fact that he was on chronic suppression. And when we retapped the knee to evaluate the enterococcus, we never saw the Klebsiella again. So that really cemented our success with the Klebsiella. But when we went back to treat the enterococcus, now with a second round of phage directed at the enterococcus, it didn't work. And so, why didn't it work that next time? I don't know. And that's one of the things that really, it's something to work on. It's a gap. What are all the things that contribute to clinical failure when you can show on the plate that that there's susceptibility, there's lytic activity.
But sort of following up again, but when John's case is presented, and as you said at the beginning, it was in many ways a huge success, in spite of the polymicrobial nature and the later problem with the enterococcus. So I guess what I'm curious about is, why was the overall result successful in spite of those issues? And what have you learned about? I mean, does that give you pause in terms of turning down other people that maybe you could help them if you had more resources?
So I think to answer your first question, John had a lot of symptomatic relief from specifically targeting the Klebsiella, because the Klebsiella for him happened to be the latest infection. So he had all of these other infections that we managed to successfully suppress. And because he was on at least 2 oral antibiotics, I believe. And those other bugs were suppressed. And then he acquired yet another infection with Klebsiella. So when we were seeing him, that Klebsiella infection was somewhat more acute. Even though it happened in the setting of a very chronic infection, that bug was actually somewhat more acute. And I think that's one of the reasons why we saw more success in that case, because subsequent cases that I started to treat were much more chronic. But sure, it does give me a little bit of pause to turn down, let's say, patients with polymicrobial infections. But I am learning that trying to reduce the number of confounders is important, because just because we, at the end of the day, there's too many factors, and there's fewer conclusions that we can come to because of the different confounders. So, in the same way that I can't really tell you how much the polymicrobial nature of John's infection contributed to the overall success or failure, it's because he had all these different factors that complicates things. So I am trying to make the cases, or select cases that are a little bit simpler.
Okay. That's interesting. So would you say that, because I was first thinking, the choice of patient, you're thinking about success rate and you want it to be successful. But I was thinking maybe there's some pressure on you as a physician to have successful cases. Will they stop letting you do this if you don't keep having successes? But it also sounds like there's a maximization of learning that you're trying to get. And that's your actual core motivator maybe in choosing a patient, would you say? Or how do you balance those?
Yeah, no, it's both. And I want to, but I think the biggest motivator is I want this to happen for the patient. because they are taking a lot of time out from their lives. A lot of them have completely stopped their lives to move to Rochester and not go to work and not see their family and live out of a hotel sometimes to do this. And it's costly in terms of time, and they also have their hopes super high. And so more than anything, I really want this to work for that person. And is there pressure on me or on phage in general to be successful? Yeah, I think so. I think there's definitely that. However, I think that we have to be very careful not to sort of bias things so that we're successful. In other words, I think we have to be careful on what we call a success and what we call a failure, and really be fair in terms of what failure and success, how they're defined. So if, for example, if you have a patient who didn't clear their infection after phage, but then had 3 more surgeries and then had the entire implant removed, infected prosthesis or whatever it was, is that really a success that we can attribute to phage therapy? So yeah, I think we do have to be mindful of the fact that, or just be as judicious and as fair as possible to what we call a success and failure. But yeah, maximizing learning too, because I don't have ready access to a phage lab. I don't, unfortunately, I am somewhat limited in the fact that I am unable to sort of, test the phage against the bacteria, multiple times during a patient's course. I don't have a lab. And so kind of being a little bit alone on a little bit of an island, it has limited my ability to kind of look at those different factors, and why I'm always sort of trying to build relationships with other phage labs and trying to create a partnership or kind of multi-pronged approach.
So our next question is related to sort of, and you mentioned PK as one thing, but sort of broadly, what sort of research do you think the community could do, either in animal models or other routes, to give you more information that will allow you to be more successful in treating patients? And you already mentioned PK, but...
Well, that's it. I mean, that's the one. PK, pharmacokinetics, I think, is one of the keys. I think that rigorous, kind of systematic animal models, but also very intentionally designed human trials are necessary, and perhaps human trials in healthy patients. So understanding where phages go in a healthy, by that I mean uninfected individual. If you give it intravenously, if you give it by mouth, if you give it, into the bladder, intrarectally, whatever, what have you, where do the phage go? How long do they stay there? What compartments do they go to? Is key. And then what happens in an infected individual? How are they different? What is the impact of the frequency of doses? What's the impact of multiple doses? And then what's the impact of the dose? The actual dose. 10 to the 10, 10 to the 9, 10 to the 11. These are questions that I think we as a field have a feeling or gestalt about, but we don't have, maybe there's not as much rigorous data around that. And I would love to be able to, look at where phages go, if there are ways to label a phage and then use advanced imaging to see where they go. That would be like a fairy tale, like a wish for me would be, wouldn't it be wonderful if I could treat a patient with compassionate use, in an individual case, give them the phage and then scan them and then, see where the phage went, and then adjust the course, according to that. that would be hard, because with compassionate use cases the way they are now, you're required to sort of, a priori give the plan, and switching and being like, oh, I'm gonna just, adjust the dose based on the, what we see on the scan. I don't know if that's going to fly, because we have to, request a change to the FDA. They have to review it, and then the IRB, the local IRB, and all of these changes take time. So that sort of real time, those real time adjustments are hard to make.
So you mentioned animal models, and I wonder if you have comparable infections that have been treated in animal models, and evidence of how different dosings have you had conversations with the FDA about that?
Well, I haven't had conversations about the FDA about that, but I know that there are good people working on it. Dr. Robin Patel at Mayo did a great rabbit model, looking at the administration of intravenous and intraarticular phage into, I believe it was phage K, into rabbits, and found that if you give phage intravenously, it gets cleared quickly, and there is a little bit of uptake in the synovial fluid as well. And then vice versa, if you give phage into the knee, then you see a lot in the knee, but in the rabbit knee, and I think, I can't remember if there's systemic absorption, but I mean, these are things that are not that surprising. But it was, I believe it was just one dose. I don't think there were multiple doses that were checked, and I don't think that these were infected. Yeah, in fact, I think I know that they were not infected rabbits.
There was one case that was, and there's probably more than this, but where the Hatful group published a study where they observed neutralizing antibodies that seemed to impact the effectiveness of the phages that they were using. And I wonder if you worry about that, and if you've ever, in the 17 or so cases that you've been involved with, have you seen evidence of that? And just your thoughts on that as a potential problem.
I guess I do worry about that. My hunch, and I'm sorry to use such an unscientific word, is that they don't matter as much, and or they're at least not a big. They don't seem to be a consistent factor, because Graham Hatful also looked at another case where neutralizing antibodies were seen, but it didn't seem to correlate clinically with the clinical outcome. I would love to be able to look at that, and I have, in anticipation of studying this, I have prospectively collected serum samples from my 17 patients in a bank, and so those are banked. I just haven't had the opportunity to, because if you want to look at neutralizing antibodies, you have to create an assay for each individual phage. And when phages are being used via compassionate use, and so because compassionate use is by nature a clinical endeavor, it's not a research endeavor. So it's been difficult to keep the phage. I'm asking other institutions to share phages with me, and so I haven't had the opportunity to collect the leftover phages, create an assay to look for neutralizing antibodies, and then test them. But I know that collecting the serum prospectively will be absolutely essential to studying that question. So they are sitting in a big bank here in Minnesota waiting to be looked at some point.
That aligns with my experience in Australia. Our -80 was filling with serum samples, and we were supposed to be, watching the phage levels and the bacterial levels in real time, and that was just so practically difficult, even though we had a lab, people, but the people were me and Stephanie, who needed to be making the phages for the next one. And so I guess, yeah, I totally feel you on just like this data is sitting in the freezer, and it's kind of an infrastructure problem to even do the learning. It's not just, you don't just learn from these cases. There's a lot of work.
Well, thank you, Jessica, for validating that, because I do feel like, there must be a way to, there's a wealth of information sitting there. But that's why we've been trying to get funding, and we've been trying to work toward, being able to do that eventually.
I also have that kind of makes me think, just like everything as you're saying, I'm wondering like on the infrastructure level, like say you had a magic wand to kind of have the infrastructure to continue to do compassionate use cases. Like let's assume there's no phage products on the market for the next 5-10 years. So compassionate use is what you have. Is it, what's your order of priorities? Is it like having phage susceptibility testing that you can do, or like somebody to take all your serum samples and give you real-time data, or the labeling that you said, like your phage is actually scannable through the body with a PET scan, like tag or whatever? What comes to mind?
Yeah. All of those. I would love to be able to have under one roof. Starting with a phage clinic, right? Oh, that would be great. If phage referrals can come see me in clinic, and then we had the capacity to see patients, and then evaluate whether they'd be a good candidate, and then be able to test the phage and acquire the phage from this under the same roof, right? So all within one institution. I mean, this is a magic wand we're talking, right? So this, if I had everything I wanted, it would be get the patient in clinic, evaluate them, take a sample, isolate the bacteria, test it against a phage library under our roof, under the same roof, and then biomanufacture it, and then have control over that, and prioritize the cases that we think need prioritization, like the patients who are not doing well clinically, and then really hone in on the efficiencies of that. At the same time, we can be doing the regulatory steps. And I have a team for that, so that's been great. And then be able to see how they're doing on the phage therapy, right? So being able to test in real time, do therapeutic drug monitoring, being able to scan them, and being able to see, where the phages are going, and then having the flexibility of using judgment to change the course if needed, just like we would with any therapeutic course in the hospital. If the patient's not doing well, then maybe we need to increase the dose or increase the frequency or change or do something else. But being locked into a certain protocol that you have to state beforehand, that you can change, but it takes a while. It's not very fluid or flexible. It would be amazing. And so that, and then having phage scientists as partners nearby, where I can walk the sample down to the lab and not have to worry about freezing it and mailing it to someplace. That would be my wish.
Yeah, just a couple of questions. I mean, you mentioned Robin, who's also at the Mayo, and Robin has done, in my mind, a lot of really nice work on trying to develop in vitro assays and seeing how well, you can get people in different labs to get the same answer with the same in vitro assay. And I wonder sort of broadly how much, when you're trying to learn from other people, and maybe your lab did one in vitro assay and another lab did another in vitro assay, and, how much is that a problem in terms of learning from other people's experiences when you're not sure they use the same in vitro assay?
I'm not sure that I understand the question fully.
Well, I mean, I guess ideally, like with antibiotics, you have you get an MIC that's done by this CLSI guidelines. You can kind of assume that the answer in Minnesota is just the same as the answer in San Diego, just picking two random locations. Um, Maybe not completely random, but maybe that isn't quite as much the case with in vitro phage susceptibility. And when you're trying to learn from the experiences of phage people treating patients in San Diego and say, well, they saw this and I'm seeing this, and you know, but are you sure you're measuring apples and oranges? I mean, not measuring apples and oranges.
Yeah. Oh, yeah. So you're asking about the kind of lack of standardization of basic assays like susceptibility testing, and we take susceptibility testing for granted for antibiotics, but with phage, that's correct. There is not a standard, and there's liquid assays, there's plaque assays, there's all sorts of different. There's user variability. There's a whole lot of variables, and what we consider to be susceptible, what has lytic activity and what doesn't. That in and of itself is problematic, definitely. So if we can't even say for sure whether or not something is susceptible, then we really don't have a lot of data to begin with, even at the start of a case. Yeah, that's definitely something I think about.
I would say, to follow on that, when you look at your 15 patients and all the courses of treatment, how often is it that it does seem like in vitro and in vivo are not lining up? Like, is that overblown that people say it doesn't line up, or is it usually the case that you see some response?
Yeah, I feel that most of my patients have seemed to respond at first. Even the ones that end up failing in the end, there's some kind of objective evidence. Either their CRP takes a big dip down and then goes back up, or there's something subjective, like their symptoms are actually better. That's subjective, so that's less reliable. But the CRP, really, the inflammatory markers seem to improve in the first week or two. That's happened in several of my patients where they've gone down. Everybody's really hopeful, and then after a couple more weeks, it goes back up, and then so too do the correlating symptoms. And so I've seen that now several times. So of the 17 cases, about 2 or so are ongoing, and of the rest that are completed, we've had about probably 50-50 success rate. And I can't. So the numerator of successes really depends on how strict you want to be with the definition of success. But yeah, it's been around 50%.
So what do you think is, for all those 50% that didn't go well, if you had to guess the main reasons for failure, like, first of all, do you think it's the same reason for everyone, or a completely different reason for each person, or like somewhere in the middle? Like, how many different factors do you think are actually important? And like, which factors are they? Is it the immune system? Is it, the phage stability? What do you think?
I think that a major factor is biofilm. So the patients who have failed kind of the hardest, if you will, are patients who have had real mature biofilms. So for example, a patient who's had a prosthetic joint in place for 8, 10 years, and they've had a recurring infection with Staph aureus or actually Staph epi, Staph lugdunensis. So organisms that can be quite indolent, but really are prone to forming biofilms. And I don't think that the phage are getting in there. They're able to penetrate the biofilm to the degree that we want. And in that way, to me, it's a problem of delivery. Because if you think about a prosthetic joint, let's talk about, let's say, a hip, and you have, or the knee, and you have the prosthesis that goes down into the intramedullary canal of a bone. So if you picture a bone as a cylinder and the prosthesis going into it, that interface between the prosthesis and the bone, like that interface, I don't know how phages would be getting in there. And so even if you are able to get phages into the joint space, that is still different than the interface between the bone and the metal. And so that I think it's a problem of delivery. So those, I would say that would be the major reason for me for my prosthetic joint infection patients who have failed therapy. Other patients, like I have one patient who has a mastoid infection. And that is with Mycobacterium abscessus. And so we tried to get to the issue of delivery by putting the phage right into the mastoid during surgery, and then putting a catheter into her ear and keeping it there for as long as possible and delivering phages right to the mastoid. And that was unsuccessful for the first several months. And now we have pivoted a little bit, and now we're really we took her back to surgery. Not me, but my colleague, the ENT surgeon, took her back to surgery. It was great because I was able to be in the OR with her and watch this whole thing, which was incredible. And I haven't been in OR in that capacity since I was a medical student, so that was cool. But I was able to see them kind of open everything up. There were parts of her ear that were totally blocked off because of granulation tissue from the infection. So I watched them really open up that space and access the middle ear so that we could get another catheter in there. And now she's able to administer phages more directly into that space. So I think for her, we have addressed the problem of delivery as best as we can. And so we'll see. She's about a month into the second course of therapy. So we'll see how that goes. I also feel that a third, or maybe second issue is just the inherent lytic activity of a phage. So Jean-Paul Pernet will talk about phage training. You've got a phage with lytic activity, and then it's trained, so to speak, to kind of enhance its killing effect. And I think that there's a factor that is inherent to the phage. So not all phages are made equal, right? Some phages work better than others, and it may not be quantifiable necessarily on the susceptibility tests that we have. So those are the reasons that come to mind first.
So just following up on something you said, the first sort of class that you mentioned, where there are these biofilms that are embedded with the prosthetic joint, are those cases where you see initial evidence of success and then a failure? Because I guess my naive thought would be that, all along the biofilms haven't been being attacked. So are the biofilms somehow later causing how are the biofilms not causing the problem initial after the initial treatment and then causing this bounce back effect? Because that would have sort of presumed that they were there the whole time and being refractory. So
I think that the initial improvement may have been planktonic bacteria that were not in the biofilm, or the free-floating bacteria that were just in the synovial fluid. In other words, I think the fact that these patients initially did seem like they were responding and then didn't even supports the biofilm idea more, because there's a component of a planktonic non-biofilm infection, and then maybe the really recalcitrant part is the biofilm infection.
right, but I guess sort of what I'm getting at, and I'll drop it in a second. But I mean, it seems like from what you're saying, the biofilm is just sort of being unaffected, but is causing a problem in a later time after you treat. But if the biofilm was there when you were seeing the improvement, why is it not causing a problem at that time and only later causing a problem?
I think it's causing a problem the whole time. It's just that the improvement is felt initially because the parts that are able to be treated, that are not in the biofilm, are treated first.
Super interesting.
I don't know that for sure, though, Joe. I mean, if I think about, the patients in whom there seemed to be an initial response, some of them are respiratory. Two patients. I had one patient who has non-CF bronchiectasis with Staph aureus, and she, in the first 7 to 10 days, said she felt better, and then like a week after that, she relapsed pretty quickly, and that was a respiratory infection. And then I had another patient who had non-CF bronchiectasis, and his CRP and numbers, came down significantly initially in the first week. And actually, with that patient, we were able to clear the sputum. He had so this is a polymicrobial infection patient. We went ahead and we treated this patient for Pseudomonas, Klebsiella, and Staph aureus, and Centrophimonas. And he received phages for 3 out of those 4 organisms, and CRP went down, and for the first time, pretty much ever, the sputum cultures had cleared. But that was very temporary, and that wasn't our goal. That's an unachievable goal, to sterilize the sputum of somebody who has non-CF bronchiectasis. But that initial signal of activity, of treatment, of efficacy was there. And then I don't know why, but he relapsed fairly quickly.
Have you ever seen evidence that the bugs are becoming resistant to the phages?
In the few cases where we've checked for that, there was one case where when we tested the phage and the bacteria again, the bacteria was no longer susceptible to that phage. Was it because of resistance, or was it because of a subisolate? Is not clear. But that was only one case where we checked where it was no longer efficacious. The other times that we've checked, it was still active.
So that's not I noticed that wasn't really on your list of failure modes or failure reasons, like resistance.
Correct.
Interesting. Well, do you want to take this to our kind of future-looking couple of questions that we wanted to ask?
Sure.
So first, where do you hope to be in 5 years in terms of your ability to treat patients with phages?
If you had asked me this 5 years ago, I would have had maybe a more optimistic answer. But I am I am more aware of the realities of all of this. And I think that in 5 years, what I hope to have are some established protocols. So some consensus in the phage world, in the world of phage clinicians about how to treat patients and different patients like we have among infectious disease physicians with other conditions, with antibiotics. I hope to have that. I hope to have more insight on PK, more maybe clinical trials. And then with the advancement of things like labeling and advanced imaging, hopefully that will play a part in that. I also would love if there was a clinical trial out there that did, more definitively show efficacy, or if it doesn't, then maybe some illuminating reasons as to why things weren't efficacious. Those are the things that I hope to have in 5 years.
And I guess at least when I was writing that question, or we were talking about the questions about where, our next question is where you're going to be in 15 years. But one of the things that was sort of, those were great answers, but I guess we were considering, where you might be in terms of, your ability to quickly find phages that match the isolate or isolates of the patient, and sort of where do you think that's going, and where do you think that's going, and what are your ideas of that?
I for that to happen, for me to be able to, or a person like me to be able to quickly get a phage, that requires a system, some sort of infrastructure, some sort of coordination. And I can do that fairly quickly because that's pretty much all I do, right? This is a major part of my life. But another physician who just is interested in treating their patient, it's a minefield out there that's very hard to navigate. I get a question from a physician maybe every week or every other week about, where do I even start? And I feel bad because I send them a long email with the steps that are needed, but I know that it's very hard to do that. So in order for a physician to, be able to send a phage somewhere and get a phage back, at least get an answer back, and then a biomanufactured phage back, I don't think that's going to happen in 5 years, at least not in the US, because there is no steps towards that right now. Jessica, Phage Directory was maybe the most coordinated effort toward that, and a very valiant one, and that's helped a lot of people. But what we probably do need is a more coordinated and centralized process, which we don't have. And so that would be like banking, or some sort of centralized database, or at least, one place where you send and you can test the isolates against other isolates, either on the plate or somehow, or virtually, like, what's the word? Not virtually. If we can use genomics, we're not there yet, but in the future we might be. So yeah, I don't think that's going to happen.
But just a question, like you sort of made it sound like because you do this more often, that you're in a better position than the physicians who reach out to you. So your process when you have a patient who you think might be a candidate is, do you have phages in house, or do you just have relationships with some companies and university labs, and you reach out to them and you have established relationships, and that allows you to do it more efficiently than someone who reaches out to you? Or
I think I just have done it, so I kind of know what to do. And I also have, I'm lucky in that I have a team of people who, well, the team is much smaller now than it was, but I have some help. And so the process is easier for me because I just know what to do. But I don't think it's easier per se in that I'm not at the top of anybody's priority list. I don't think necessarily. I know that our friends at Baylor have really, helped us a lot, but I appreciate that so much. But I think it's the same onerous process, but it's probably easier for me simply because I know what to expect and I've done it now several times. But the process is still long and arduous. It's still, I have, so I get the isolate, send it to a lab. I have relationships with university labs. My commercial relationships have with phage companies that do that. Are no longer, in existence. So it's really academic centers like Pittsburgh, Daria Van Tine, Graham Hatfull, Anthony Moresso, Austin Terwilliger, Kiko Salazar at Baylor, and then David Pride and I have also been talking. David Pride at UC San Diego, and so those are the main folks that I reach out to when there's a patient in need. But if you notice, I've treated 15 patients, 17 treatment courses in 5 years. That's not a lot.
right.
And so part of the reason why it's taken so long is that it's a slow process, and it's a lot of work.
Yeah, and I think one of the things when Jessica and I were kicking around various things, a former colleague of mine at NIAID, when I used to talk about some of these issues, I guess this is a little bit off the wall, but there's this repository of anti-venom that's run by the Florida, I forget, wildlife, that because people get bitten by poisonous snakes in Florida, and either the county government or the state government keeps this stock of them. And I guess sort of you can imagine a situation where the government might get involved in this. And I know there is some interest, I think, as I mentioned to you at some point, in doing this from some people at the NIH Clinical Center. And, I wonder how you think that would look.
Yeah, I think so. I love that idea, Joe, because what that program, that anti-venom collection is prioritizing is patient lives, right? So this collection of anti-venom was put together for one purpose only, and that was to save the patient who gets bitten by a snake, because snake bites are everybody's problem, right?
Right.
And so the notion that antimicrobial resistance is everybody's problem, it should also be, that's also everybody's problem. And so if there were a similar collective response where, the phage that had the most lytic activity and the broadest host ranges were sitting in a repository in some central location, that would be amazing, right? But it's not quite working out that way, because these are these are people's phages. They're carefully curated and collected phages that can sometimes represent a person's lifetime of work. And so, it's hard to, give those up into a repository. So there's a lot of conflicting, priorities, and I would love it if we treated antimicrobial resistance and difficult to treat infections like we treat climate change, for example. It's a problem for everybody, and we need to centralize efforts to find a solution. And a big payoff profit may not come with that. Maybe we ought to think about that.
Yeah, I mean, it is. I always think there is a big similarity between AMR and climate change, and the fact that, as opposed to something which was obviously horrible, like COVID, where a bunch of people are dying in a big group, AMR is just sort of, unfortunately, slowly killing people at a steady but appreciable rate, but it's not quite as dramatic as COVID that gets everyone activated in a way that maybe is commensurate with the degree to which it's a problem. And climate change has similar aspects. So, you mentioned being at Stanford, and I happened to be in the Bay Area after one of the big fires, and it looked like Mars or something. The pollution. This was 90 miles away, so I mean, it's obviously having huge effects that we can't even really determine the overall effect.
I do think that having centralized biobank or phage bank, or at least access to or centralized process would be very helpful in kind of personalized medicine. The Madrigal system for regulation of personalized cocktails, like they have in Belgium, is magistral system is something that we could maybe learn from. But I do think I am hopeful, just to be hopeful here too, and optimistic. I truly am hopeful that we will have at least one. We will start to have some fixed cocktail products coming onto the market. And you mentioned that, you were going to ask the 15-year question. I think in 15 years we will have that. Probably a fixed cocktail will, be available first. I'm going to guess that it's going to be a UTI type of indication. And that's going to be very helpful. And that doesn't mean that there's no room for a personalized approach. We don't have to pick one or the other. It will be both fixed cocktails and personalized approaches that will be needed. And then things adjacent to phage therapy too, like endolicense. It just in the same way that overcoming climate change will take solar and wind and geothermal and other things, we will need all of those different modalities to tackle, to address, to solve this antimicrobial resistance problem.
Here I thought phages were going to solve everything. No, sorry.
Yeah. I love that. And that's a really nice way to kind of close out with a little bit of hope. And I guess I also am glad that you touched on the cocktail side, because that was going to be kind of my other question, is because I completely agree that we're going to need both prongs, and both of them, they have separate roles and they're both great for certain sides. And I don't see a future where we don't have both. On the cocktail side, one of my burning curiosities that I hope we can find out the answer to in this series, we can't finish it until we have some satisfying answer, is why have all the clinical trials been failing for cocktails? Is it because of phages, because of cocktails? I don't really think it is. Or could it really be that just it's people problems every single time? Or do you think it's more about what you're talking about with the delivery and the PK? We have so little window into the in vivo reality that the design has just been too hard for people. what is the best answer that you have for these failing clinical trials, and is it the phage's fault?
Yeah, I know. For a long time, I chalked it up to the lack of personalization, and I still think that may be a large part of the problem, because the trials that are taking place now, that use fixed cocktails, they're not performing. Not all of them are performing susceptibility testing prospectively, right? So I think that's a big problem. That's the big thing. And then kind of the PK and the other things. But these I think the cocktails seem to be quite good. But is it right for all the patients that they are being tested against? And the numbers of patients are still quite small, so ends of 8, ends of 12. Is that enough information to say this fixed cocktail that we didn't treat susceptibilities didn't work in these 8 patients, or didn't work in the majority of these 8 patients? I don't know that that's enough information. So I still chalk most of it up to the lack of personalization. But then that calls into question, the whole premise of fixed cocktails, and would that be even feasible and marketable and successful in the marketplace? So yeah, it remains to be seen. Yeah, I wish I had a better answer.
No, I think it's good to. I mean, I like the fact that you are quite willing to say, "I'm not sure" when you're not sure, because that's almost. I sometimes don't trust people who claim to know the answer to everything, because then I feel like they might not be. They might have misplaced confidence.
Well, nothing will humble you like phage therapy or phage.
Yeah.
We had a last question. Are there scientific, clinical, or regulatory or economic challenges that you see preventing where you'd like to be in 5 and 15 years that we haven't touched on? Because I think we touched on many of those challenges during the course of your thoughtful answers. But is there something that you can think of now that you didn't touch on that you'd like to highlight?
I would maybe just emphasize that I am sometimes amazed at the trouble people in this field are having with getting funding, right? And it seems to me that all of the other sophisticated medical breakthroughs that also suppress the immune system, like CAR T therapy, for example, and all of the sophisticated and life-saving cancer therapies, all cause or can cause infection as a side effect. I wish there was more of an acknowledgement, or that infection is not just its own thing. It's integrated. It's everywhere. It not only it impacts cancer patients, it impacts every field, and with every new technology or development, a new thing that gets put in the body, another device, that's another problem, that where infections are the problem. That's another potential for infection to be the problem. And so I don't know if I'm articulating this well, but I just wish that the people who make policy decisions and decisions about where the money goes realize that CAR T is not going to be helpful if your patients all die of infection.
Yeah, I mean, that's an issue that, I used to go when it was in Washington. I would go to the World AMR conference every year, and people would discuss this issue and how, there's a couple factors that I think we're sort of all a little bit aware of, that, people, when they get infections, they tend to, either get treated and recover, or sometimes horribly die fairly quickly. And so there isn't this chance, like with cancer patients, where there are these people who are suffering, unfortunately, with cancer for like 5 or 10 years and build up support groups and lobby together. I mean, I guess it isn't as great a venue for getting like support groups for people with AMR. I mean, you have CF patients who have lobbying for AMR, and but you sort of highlighted a strong reason for doing it, but you know you have a diverse group of people who are being treated for a diverse group of things, and AMR is in common with all of them, but they're not like banded together in the way that cancer patients band together, because they all have, I mean, they have different kinds of cancer, but they all have cancer, or people who need implants have common issues, but then they have AMR. But the cancer patients and the implant community and the CF community don't all get together as an AMR community. And I don't know how to, other than banging the drum, how to get that done. In terms of funding, I think I've mentioned to you that one of my former colleagues at NIAID has just put out a call for that's coming out soon for center grants and phages, and there's also centers to treat AMR. And I guess Jessica and I were talking about this a little bit when she actually interviewed me. But do you think that the ability to apply to NIAID and other institutes, that the prejudice against phage in study sections is getting less of a problem, or do you still find that phage modalities of AMR treatment are still maybe getting not as good a chance to?
Well, until you actually said prejudice against phage, I didn't actually think that was happening, but maybe it is. My perception, and to be honest, is that I feel like it's worse in the lab. I don't know. I mean, that RFA for the phage centers is a bright shining light in all of this, and I'm so glad that that's there. And I'm really hopeful that there could be, that's the start of something big for the field. But but everyone I know in my personal world of phage people, like, no one's getting any funding. And so, yeah, that's just my perception. I'm sure, I don't know the real numbers, but I feel like it's gotten worse over the past couple of years.
It'll be interesting to see. I mean, I guess, there's NIAID is also putting out sort of a general call for AMR approaches around the same time as they're putting out the phage specific call. And, I mean, I guess it will be interesting to see if people put in phage applications to that, if they, there's still a prejudice in study sections that aren't set up to review phage grants to.
Yeah. Well, the fact that it's phage specific is,
Yeah, well, yeah, one of them is and one of them isn't. So
yeah, that's amazing. And what a great thing.
yeah, no, I mean, there's people who are thinking about it a lot, obviously, but it's hard. But I don't know, Jessica, do you have more questions, or should we thank Gina for her time and very thoughtful responses?
Yeah, I love this. Thank you so much, Gina. And Joe, thanks for making this happen. And just, it's so much nicer to have two interviewers, because I can like think and have like way more time to think of my questions. But yeah, this is so fun, and it sets the tone for the whole series. And it's going to be so interesting to see if we kind of ask a similar set of questions to a bunch of people, are we going to get the same answers? And yeah, I know that all these things have been flying around and people have put them in review papers, and we're Gina and I are writing a review together right now, which is coming to your inbox soon with the final edits I worked on last night.
Oh, God.
I mean, it's like I feel like it's been a swirl of these concepts. And so I'm trying to pluck out something new or some new synthesis, or at least some like 2024 synthesis of where we're at. And I do think it's shifted a lot. And to be honest, I never really read any of the reviews anymore because I'm like, there's another one, and I should, but it just becomes this whirlwind swirl of all the same keywords. And I don't know. I think that's why we're talking to people, because you just need to ask the questions point blank, like, why is everything happening this way? Or are we making progress? And I think you got both sides really, really well illuminated. So thank you so much. Joe, do you have any parting words?
Well, I think Gina was very kind to the two of us and said that we were some of her favorite people. And I think it's probably an obvious reflection of how we feel about you that we picked you first, because you are, I think it was evidenced by what you said here that you're someone who obviously cares deeply about your patients. And, obviously I imagine there's nights the last thing you're thinking about before you close your eyes is what you should have done or could have done. And the first thing that you're probably thinking about when you wake up in the morning is what you should have and could have done. And so thank you for doing that. And thank you for just, I think, you and there's a few other obvious people who just are just, making the phage field an open and wonderful field to be a part of. So I think Jessica and I both feel lucky to be part of it in some way.
Thank you. That's so kind of both of you. Thank you so much. It was an absolute pleasure to talk with you, and let's talk some more.
Yeah.
Whether or not there's a mic and a recording device.
I have so many ideas.
Yay.
Yeah.
All right.
All right.
Thank you. Bye, guys. I'll see you soon.
Thank you so much. Bye-bye.